Yeah. Okay. Thank you very much for all of you that are attending this webinar. Today we have the great pleasure to receive at Unicamp Professor Lewin, that is Professor of Ecology and Evolution at the University of California, Davis, and member of the U.S. National Academy of Science. There are many other skills of Professor Lewin, but one of the most important is that he's currently leading the Earth BioGenome Project, a global coalition aiming at sequencing, cataloging, and categorizing the genome of the Earth's eukaryote biodiversity by 2028. Ebp is one of the most important developments after the Human Genome Project has been completed, and technology generated by this project and further improved has boosted innovation in health, energy, and food security. In this talk today, Professor Lewin will discuss about the Ebp in the context of the climate and biodiversity crisis. Professor Lewin, thank you. Lewin, thank you very much for visiting us at the Unicamp, and the floor is yours.
Thanks very much, Paulo. It's really great to be back in Campinas, even though we're still in the middle of the pandemic. I do have my mask here. And I have to say that it's great to see the people who have come today despite the pandemic, and I really appreciate the invitation to visit the campus despite the conditions that we're all working under. And so the last time I was here to talk about the Earth BioGenome Project was 4 years ago, when the project was very new and we hadn't had an official launch yet. And so now, 4 years later, I have a lot of progress to report, and that will be the subject of my talk today. So our understanding of life on Earth is really limited. Even at this point in the 21st century, we still know maybe 9% of the species that exist have been named by humans in the classical Linnaean taxonomic system. So of the estimated 12 to 15 million species that exist, only 1.8 million have been named. So that's approximately 12 to 15%. So there's still more than 85% of biodiversity that has yet been undescribed by men and women. And the reality is that this life on Earth, is under existential threat at the present time due to changes in land use, overexploitation, invasive species, and of course, most recently, it's been recognized that climate change is one of the greatest threats to biodiversity on Earth. Of the 137,500 species that have been plant and animal species that have been surveyed by the International Union for Conservation of Nature, IUCN, 28%, 38,500 species have been shown to have their population size severely reduced and are under threat of extinction. And we also know that in the last 100 years, 543 species have been termed to be extinct. And this rate of extinction is 100 to 1,000-fold greater than the background rates of extinction, as has been determined from looking at the geological record. And so by the end of the century, it is projected that 1 million species of plants and animals may become extinct if we do not change the way humans are impacting the planet, either through land use change, overexploitation, and climate change, pollution, invasive species, and such. And nobody knows if we reach that 1 million species becoming extinct, nobody understands really what is going to happen on Earth. This dramatic increase in extinction rate has spurred great efforts now across the planet to conserve and preserve biodiversity, because the tipping points are really unknown. The tipping point at which we could have complete global catastrophe, collapse of global ecosystems, based on because of the loss of biodiversity. So loss of biodiversity not only threatens biodiversity, but it threatens then us as a species. It threatens our societies, global political stability. Okay, and this is why there is so much now concern and activity in the area of biodiversity conservation, and that's because biodiversity itself is the basis for ecosystem services, those quote unquote services that are provided to us free of charge by nature. For example, water purification, agriculture, medicines from nature, regulation of the biogeochemical cycles, and so on. These are the ecosystems that biodiversity supports, and if we lose them, we could be in a very precarious situation as humans, and support for our societies and our world. And not only is this important, again, for the reasons that I mentioned, but biodiversity also plays a critical role in the economy of the world. So more than $400 billion just in the US and in Europe alone in 2017. So probably today it's 500 or 600 billion. There are no more current figures than the ones that I'm giving you today. But the global bioeconomy is very much dependent on biodiversity as well. And so this is extremely important. And today, with all of this happening in nature, there's now this increased emphasis on trying to preserve and conserve biodiversity where it exists in the world. And perhaps one of the greatest tools that we have in our toolkit for conservation of biodiversity is genomics, and understanding of the genomes and development of tools for conservation, as well as for understanding adaptation to the changes in our environment that are occurring as a result of climate change. So genomics is just one. It's not the only tool, right? The big one that's being addressed in Kunming, by the Convention on Biological Diversity, setting aside 30% of the land and 30% of the oceans to try and preserve at least 50% of the species on Earth. But there are many problems, as many of you can anticipate, with such a strategy. Which 30% do you preserve? What species are expendable? Which species are not? If we're going to have a very active, managed conservation program, genomics is now an extremely powerful tool to do so. So what is the global status then of eukaryotic genome sequencing? I'm not talking about archaea, I'm not talking about bacteria here. There are probably trillions of bacterial and archaeal species. We're going to focus our conversation on the eukaryotes. So as of just a few months ago, this nice rainbow chart, which was produced by my colleagues at the Sanger, Wellcome Sanger Institute. They're called the GOAT team, which is Genomes on a Tree team. And we all work together in terms of aggregating the data across the EBP to understand where we are with respect to global biodiversity sequencing efforts, as well as Earth BioGenome Project efforts. And if you look at this this chart, this rainbow chart, you'll see that the numbers are still quite sobering. When I was here last time, I probably told you less than 0.3% of eukaryotic species on Earth have had their genome sequenced. Well, it's doubled, in the last 4 years, but that number is still 0.6% of known species. And if we only know 12 to 15% of the species out there, we still have a genome sequence for less than 0.1% of total eukaryotic biodiversity. So everything we know at the genomic level is based on just a small fraction of the biodiversity that exists on Earth. So just imagine what we can learn and what we can do if we have more than just 0.6%. And this is part of the rationale then for the Earth BioGenome Project. And if we look at it and say, okay, we have about 0.6%, that's nice. How are those species distributed across the different taxonomic groups? So maybe it's no surprise then that the greatest number of species that have been sequenced are the ones that most people would be working on, the plants and the animals and the fungi. But as you can see, these numbers are still paltry. They're very, very small. And what's even more interesting, of course, is when you separate Linnaean taxonomy and you look at phylogenetics, all of those groups now, all of these different groups, these what are called now supergroups, are in this only 469. So most of what has been sequenced, okay, is plants, animals, and fungi, things that we can see or things that most people work with, but the vast eukaryotic biodiversity that exists in nature are in these single-cell microbial eukaryotes and in the very small animals that live in the ocean and in the soils, okay? So this is unexplored. And so for all of the graduate students and postdocs that are listening, this is a tremendous scientific opportunity to study the vast biodiversity that exists at the genomic level, if you can get your hands on the organisms and obtain a sufficient amount of DNA to sequence. So, given this great challenge of less than 0.1% of biodiversity that's had a genome sequence, a complete genome sequence, and this vast biodiversity that's completely untapped, several years ago, we proposed this project called the Earth BioGenome Project. In 2015, the idea first came. In 2018, a paper was published in PNAS that describes sort of the manifesto for this project. And the goal of the Earth BioGenome Project is to sequence the genomes of all 1.8 million known species of eukaryotes in 10 years. And the Earth BioGenome Project then is a organized, confederated international network of networks that has been assembled to accomplish this goal. And so, okay, so we have this goal of sequencing everything. So the ultimate goal, of course, is, at least from the point of view of genome sequencing, is to obtain a digital repository, meaning the sequences of all of eukaryotic life on Earth. Why would you want to do that? You would want to do that so you could address fundamental scientific questions that cannot be addressed by sequencing a few organisms, less than 0.1% of the organisms on Earth, or even a small representative group of a single taxon. These are the kinds of questions that are shown on this slide, that can be addressed only by sequencing all eukaryotic life. This is the only way you can understand the full expression of the tree or trees of life. And so approaching this from a very systematic phylogenetic approach will give us the answers then to deal with a number of important problems in a practical sense that are facing us from this loss of biodiversity and these changes in our environment that are resulting in this rapid loss of biodiversity, such as improving agricultural yields and facilitating climate adaptation. This is what we're talking about here, actually in Campinas, with the fantastic project that Paulo Arruda is leading. And also building security, pandemic security, enhancing human health. I'll talk a little bit about some of our own work from my laboratory a little bit later, supporting ecosystem services, and of course, mitigating the effects of climate change on biodiversity. These are some of the applications that will come from answering these very fundamental questions that can only be addressed by sequencing everything. Okay, and if you're interested in this, of course, I mentioned before the 2018 PNAS paper, but in January next month, there'll be a special feature published in the Proceedings of the National Academy of Sciences of 10 papers on the Earth BioGenome Project, and covering all aspects of the project, many of which I'm going to cover in the remainder of my talk, but also the ethical and legal and social issues around the project, as well as issues relating to bioeconomy and indigenous rights. And I think you'll all find this very exciting when it is published, probably the middle now of January. I just learned yesterday that all the papers have been, all these 10 papers have been accepted, so we're well on our way to having this special feature published. So what about the progress? When I was last here, it was just a few laboratories involved in this. This effort has now grown to 43 institutions and 49 different affiliated projects. Those are related to specific eukaryotic taxa, involving 5,000, at least 5,000 scientists around the world, and 22 countries. And you can see on the map, of course, is Brazil. Why is Brazil on the map? Because FAPESP is an institutional member, and that covers all of the Sao Paulo institutions that FAPESP funds. And we are looking forward to even greater participation from FAPESP, of course, in our international efforts. And I'll point out another feature. Just in the last year, we have the Africa BioGenome Project joining us. This is a really critical project because, you know, a significant fraction of Earth's biodiversity exists, endemic biodiversity exists in the African continent. This project happens to be centered in Kenya, but involves 22 different countries in Africa right now. And altogether, I mentioned 22 countries. That's where the projects are headquartered. But if you look at the number of countries that are involved in the Earth BioGenome Project, it's now more than half the countries of the world, more than 100 countries. And if any of you, I don't know how many of you attended the Biodiversity Genomics meeting this last fall, more than 100 countries and more than 3,000 participants. So this project is really capturing the imagination of the world, and it's now become sort of like a tsunami, has a life of its own. And it's, for me, it's very exciting and gratifying to see this happening across the world. And so our model is this hub and spokes model. This is just a model. It doesn't show all of the organizations involved. But if you look around the outside spokes, those are the major regional sequencing and informatics labs. Around the inside spokes are all of like those 49 projects that deal with specific eukaryotic taxa. And the Earth BioGenome Project, the thing that I chair as the international coordinator, is this central function that deals with development of standards across the entire project. How you collect samples, what are the standards for sequencing, what kind of metadata collected with each standard, what types of analyses, standardized methods and approaches for approaching the comparative analysis, and coordinating communications across the entire world for this project. And all of that is headquartered at the University of California at Davis, where the secretariat is located. And the project itself is committed to open access of all data in compliance with the Convention on Biological Diversity and the Nagoya Protocols on Access and Benefit Sharing. This is an extremely important part of what we do, extremely difficult part. I was talking to Paulo over lunch about how critical this component is, because without benefits, there's no access, and without access, there can be no benefits. So it's a really important a part of the project, and we have many people working on this particular component. We're also committed to diversity, equity, inclusion, and justice, environmental justice throughout the world. The EBP is designed to be as inclusive of as many scientists around the world for the benefit of all peoples of the world. And those are not just words, okay? We have a very well-established committee structure to deal with things. We have a Jedi, like the Jedi Knights committee, which is chaired by Sadie Paez at Rockefeller University, and Marcelo Uliana Silva, she's a Brazilian, who is at the Wellcome Sanger Institute, and she is co-chair of our Jedi committee. Our Jedi committee has representatives from 11 countries throughout the world, and this committee is really just getting going with its activities this year, and you'll be hearing a lot more about their activities as the project unfolds. We also have an Elsi committee. This is built in right from the beginning. Elsi is ethics, legal, and social issues. We have representatives from 4 countries, including Brazil. Again, Manuela da Silva, who many of you know, she's at Fiocruz. She's on this committee. They have a very significant paper that is coming out in that special issue on Elsi issues that are very specific to the Earth BioGenome Project. And I think you'll all be interested in reading this report when it comes out. And of course, we have the International Scientific Committee, which is chaired by Federica De Palma, who's at Genome Canada, also the University of East Anglia in the UK, and then 5 very distinguished scientists that chair these different standards committees. Meryl Lonachak from Wellcome Sanger Institute, Sample Collection and Processing. Richard Durbin, who's very well known for the Human Genome Project, chairs the Sequencing and Assembly Group. Paul Fliczek from the European Bioinformatics Institute chairs our Annotation Committee. Shustin Lindblad-Toh from the University of Uppsala and the Broad Institute chairs our Data Analysis Group, and Xiaofeng Wei from BPI chairs our IT and Informatics Group. So this is all on their way, and in that special issue that I talked about, the Standards Committee has a report as well. And if you go to the Earth BioGenome Project website, you can already see the recommendations that have been made by each of these subcommittees and for standards for everyone in the project to follow. It's very important that we have standards, because if we have 49 different projects following 49 different protocols for everything, at the end we'll have to start again and do everything over. So we don't want to have to do this again. Okay, and the Earth BioGenome Project, and how is it organized? Well, there are really 3 phases. We're just completing now the pilot phase, and we're moving into phase 1, which is sequencing a representative species of each of the known eukaryotic families. So there are about 9,000 now, 9,400 families, and we're going to make a reference quality genome, the highest possible quality in terms of contiguity and base calling accuracy that we can make. And those recommendations for producing reference quality genomes are in this report, in the report I just mentioned, and on the website. That's phase 1, first 3 years. Next 4 years will be a representative species for each of the known eukaryotic genera, and the final phase is the rest of the species on Earth, out years through years 8 through 10. And that's the one strategy that's being employed. The second strategy that's being employed, and this is being done in several locations, is this ecosystem sequencing strategy, where you go into a particular place on Earth. It could be a long-term ecological observatory. It could be a forest or an area where there's been monitoring for long periods of time, data collected on large numbers of species, and sequence everything in that particular environment, okay? And that would include in the soil, the land, the water, in the air. And in this way, to build a basic knowledge of a particular ecosystem or ecosystems, build models from the ground up so that you can then monitor over time. Just sequencing one organism from one environment is not going to help you very much in understanding how climate change is affecting biodiversity. But if you first characterize every species in a particular ecosystem, you can then develop the tools, for example, eDNA monitoring systems and biosurveillance mechanisms to follow changes over periods of time as a result of change in the environment. And in that way, to produce this multidimensional and dynamic view of all of life on Earth. Okay, so you saw this figure before. This is the current status of sequencing, pretty dismal, maybe 0.6% of species described. But look here at the highest taxonomic levels. 80% of that, not even a representative species at the phylum level has been achieved. Okay, and it just gets lower and lower as you go down through class, order, and family, and so on. So as I mentioned before, the first phase is sequencing a representative species from each family, and at the current time, there's a genome sequence only for about 18% of the families, and we're going to talk a little bit more about that. So what about the EBP project? Well, EBP is only going now for 3 years, but within this total, about 9.3% of all species now can be attributed to the EBP, with those 49 EBP affiliated projects. So just in a few years, EBP is already having a significant impact. And if you look at how genome sequencing is going across the world, and these again are whole genomes, not a mitochondrial genome or a sequence tag from, this is whole genome sequences. You can see that over the past 3 years, from 2019 to 2021, the number of genomes has doubled in just a few short years. That's great news, but the not so great news is if you look at the quality of those sequences, the quality is not sufficient in most cases to do the kinds of studies that we would like to do if we had complete, gapless, near-perfect assemblies. And so these sequences here, the rose color and the orange color, these are the chromosome scale assemblies and the complete genomes, and the rest of these genomes are fragmented. These are scaffolded assemblies or contig assemblies, which have many gaps and many errors in their order and orientation of scaffolds. So only 15.4%, even though we have more than 8,000 species now with sequences, only 15.4% are of sufficient quality to meet the EBP standard. And if you look at the EBP's contribution to those high quality chromosome scale assemblies, okay, so this is only since EBP came online in the pilot phase. EBP has already produced about 800 now whole genome sequences, and look at the rose color here in the last year. EBP, of all chromosome scale assemblies that exist in the world, the EBP has now produced 40% of those chromosome scale assemblies. So we're making a huge impact, and we're just really getting going. And these are the kinds of assemblies you really need to do the kinds of science that are going to be necessary to answer the kinds of questions that you're asking here and that others in the world really want to address as well. And again, this is another look at the eukaryotic families. So this is the whole world, right? There are 1,589 eukaryotic families that have a whole genome sequence, but only about 30% of those are chromosome scale, okay? And if you then look at the EBP's contribution, here it is again, because you can see again EBP's contribution on chromosome scale assemblies, 26% of all families, so that's a much higher fraction than the total. We had 9.3% of all species, but at the family level, it's now 26%. And 40% of all chromosome level assemblies at the family level are contributed by EBP. So this is really starting to have a big effect. And this is an underestimate. It's really over 50%, because these are only the projects that are accounted under the EBP umbrella. Project ID, this PRJNA533106. And we're not capturing all the data yet in the world. For example, from JGI, who has 200, they have their 1,000 Fungal Genomes Project, and they have, two representative species from 100 different fungal families. Okay, and then you have the BGI, which are not in ISNDC, so it's actually way higher than this now. But the next time I come and present the data, we'll have all this data properly aggregated. But we're just able to do this now because people are using our umbrella project to identify their sequences. And if all goes well, by next year, if we have all the commitments that people have for delivering sequences to EBP, we're going to have 3,000 reference quality sequences delivered. That's based on current commitments from these 49 projects throughout the world. And most of these will be at the family level. So at the end of the first really complete year of the project, remember I said 9,400 families, eukaryotic families. We're going to be almost on target at the end of this year. We'll have approximately a third of the families by the end of 2022 in public domain databases as a result of our efforts. Whoops. Okay, and so the papers are starting to come. It's really fantastic to see. Last April, the first paper from the Vertebrate Genomes Project, which was critical in establishing all of the standards for genome sequencing and assembly for the EBP. The first 16 were published in one issue of Nature, proving for the first time that reference genomes can be produced at scale. It hadn't been demonstrated before. These were the first 16. Now, in this year, they're over 100 from the VGP alone. Okay, and these are big genomes. These are 3 gigabase, genomes. So they're much bigger than many of the plant genomes that you're working with and insect genomes, for example. So all of this is under the leadership of Eric Jarvis at the Rockefeller University. And in addition to those papers, of course, describing how the sequences were created and the assemblies were done, there were a series of papers that were published with this paper describing the biology that reference quality genomes could enable. That's really what it comes down to, is what kinds of questions can you answer? And so I urge you to look at these papers because they cover everything from conservation to chromosome evolution to memory and cognition. Now, when you go in and look for a gene, you don't worry that the sequence, is wrong or that you're missing exons. You're sure that the gene is there, and you're very sure about the accuracy of the sequence, because one of the requirements is, in terms of accuracy, is less than 1 in a million errors. So that's being achieved routinely now by the project. Another big project is the Tree of Life project, funded by the Wellcome Institute, Wellcome Trust at the Wellcome Sanger Institute, that's led by Mark Blackster. It's the largest team in the world right now doing biodiversity sequencing. They have already produced and submitted 210 reference quality genomes, not fragmented. These are all reference quality genomes, and have 1,300 genomes currently in progress, and spread across the entire phylogenetic spectrum, as you can see here. So these projects are moving quite rapidly. Of the 3,000 then reference quality genomes, just one laboratory alone is producing about a third of those. And one of the real missions of the EBP is to spread this technology throughout the world, is to develop capacity in the developing world where endemic species are located, so that we could rapidly scale up and not miss critically important species. Another fantastic paper, this was done with a more with short read assemblies, was the paper produced by the Zoonomia Consortium led by Ellen Carlson at the Broad and University of UMass Amherst, and Justin Lindblad-Coe, who I mentioned before, who's at Uppsala University and University of Massachusetts. And they produced 131 new vertebrate sequences, mammalian sequences, sorry, as part of their project. And what's shown in this figure are 7, if you look at the named species in the red triangles and dots, these are 7 species that are on the IUCN critically endangered list. And you can see from this figure that there's a relationship, even from sequencing a single individual, you can gain insights as to which species may be threatened or endangered by looking at just the runs of homozygosity, that's stretches or runs of homozygosity and overall heterozygosity. So species that are on, of the 131 species, most of the 7 species have low overall heterozygosity and long stretches of homozygosity in their genome. And this is very valuable information just obtained from a single individual with short reads. Another project that I'm going to spend a couple of minutes talking about is one that I'm involved in Zoonomia. I'm also involved in the California Conservation Genomic Project. This project started a little over a year ago. It's led by Brad Schaefer at the University of California, Los Angeles. And this is a project that's a large-scale collaborative effort in California to conserve flora and fauna in California using conservation and landscape genomics of threatened and endangered and commercially important species. This project involves the sequencing or resequencing of 225 different species organized in 130 different sequencing projects involving 79 investigators across all 10 University of California campuses. We are producing 150 reference quality genomes for 150 different species using the same strategy as we use for Velocia here, which is Hi-Fi, PacBio plus Hi-C. Sequencing work is being done at the UC Davis Genome Center, and the OmniC technology is being done at University of California, Santa Cruz, and at Dovetail, a private company located in Santa Cruz. We're also doing annotation of all of these genomes, which is part of a program called Illumina EBP. Illumina is supporting the Earth BioGenome Project, and it's providing the reagents to us for annotating 100 genomes of plants or animals that are endangered or threatened. And there are going to be 150 resequenced genomes. So we're going to be doing population level genomic diversity for each one of the species that we have a reference genome on. Getting close, good. And so 20,000 resequenced genomes, and the number of populations in each area of the state of California is shown here. So we're really covering the entire state, and the goal then will be to develop very good management plans from working with the California Department of Fish and Wildlife to develop management plans for the conservation of these species in the state of California. And as you can see, the species diversity is extremely high. All plants and animals in this case, we have 100 unique families represented among these 150 reference genomes across vertebrates, plants, and plant species. Okay, and we're doing quite well. We have all the species targeted for reference genomes already collected and sequenced. I won't go through all this, just a progress chart. 15 genomes are already completely assembled, and we're moving quickly. We think by the end of the year, we'll have all of our 125 reference quality genomes for this project completed. And then what other applications I mentioned before, improving pandemic prediction and responsiveness. This is a paper which was produced during the pandemic, which I led, my postdoc, Joanna Thomas, and then an international group worked on this particular paper. It's received, as you can see, enormous attention, alt metric score of 2080, and you were mentioning citation, already more than 250 citations in a very short period of time. And it demonstrates what you can do when you have an open, freely accessible database of sequences, because we realized once the receptor for SARS-CoV-2 was known, right, which is the ACE2 receptor, that we could go to the public domain databases and fish out all of the ACE2 receptor sequences. And we were able at that time to get 410 unique vertebrate sequences, and then use the human receptor, okay? And because the three-dimensional structure, and it was known which parts of the ACE2 receptor that SARS-CoV-2 interacted with on ACE2, we could then develop maps across all of those species ACE2 to be able to predict, using structural modeling and homology modeling, to predict which species across those 410 species would have the ability of SARS-CoV-2 to bind to those species ACE2. And we also did evolution. We looked at where the accelerated regions were and which were the regions that were under selection in ACE2. So it's a long story. I can't tell you this whole story today, but you can read the paper. The very interesting outcome for conservation is that we could predict species in different groups, which we predicted would have, where the SARS-CoV-2 spike would have the ability to bind to ACE2. And of course, very soon within the pandemic, all of the non-human primates, they confirmed. We had the gorilla in the San Diego Zoo, and chimpanzees and rhesus. Basically, all non-human primates had 100% identity across the group of 25 amino acids that were shown to be critical of the binding of ACE2 to SARS-CoV-2 spike. So no surprises there, But then in the high group, we said, wow, there's some very strange species. Sure enough, Chinese hamster and Syrian hamster confirmed. And I don't know if you saw the reports just 2 weeks ago on the white-tailed deer. That's a ruminant. You wouldn't predict that based on the phylogenetic relationship. 60% of the white-tailed deer in the US are infected with SARS-CoV-2. And so these were important. Two reasons. One, spillover from the human population into wildlife, so very important conservation issues, and then spill back from wildlife populations potential into the human population. Because why? Why did we do this? Because the intermediate species for SARS-CoV-2 is still unknown. We don't know if the virus jumped from the bats to an intermediate species to humans, or whether it jumped from bats directly to humans. It's still unclear. The intermediate host is still not identified. And so this list provided in April, really, when we put it up on bioRxiv, was published later in the year. This list provided a list of target species that one could look at to see where the reservoirs for SARS-CoV-2. And it turns out most of the predictions that we had made have been accurate where they've been tested. Okay, so to conclude, we know that sequencing of 1.8 million eukaryotes is not trivial. There are many obstacles in getting there. We know it's not going to be easy, but we're going to work very hard at doing this. And we know what the challenges are. I think I don't have to go through this in detail because I'm much out of time, but I will say that we have the technology today to achieve the 9,300 and the scale throughout the world. So phase one, we're going to make it in 3, maybe 4 years. Phase two, we have to increase our output by 14-fold to get on a daily basis in the world. We're going to have to be producing 123 genomes a day, every day around the world to get to 180,000 genomes in the 4-year period. And of course, to get to the next level, we got to go 10-fold above that to get to 1.8. So there are many challenges that we have. We recognize them. But we would not have predicted 4 years ago when I was here before that we could even get to these highly contiguous genomes that we are at today. And we know there are going to be major advances in throughput in the next few years, even with the existing technologies that we have today. So sourcing, acquiring, this is the biggest problem, right? The technology will not be the problem. Getting samples and feeding this pipeline with high quality, high molecular weight DNA from vouchered specimens so that you can always go back if you need to. This is going to be our biggest challenge. Automating this up to get to 1,000 genomes a day is going to be a huge challenge. Assembly and curation, we don't have any of these tools to do this at the scale that will be needed to get to phase 3. And then thinking about tools for analysis, comparative genomic analysis, we can do only maybe 100, species, but to scale up to do genome alignments of thousands of genomes, lots of scientific challenges lie ahead. So, but we can't quit. E.O. Wilson, of course, tells us that we have to work hard at this because every single scrap of biodiversity is priceless. If we lose a single species, we lose the collective intelligence of millions or hundreds of millions of years of evolution. And we don't want to do that. And of course, once a species goes extinct, extinction is forever. So our action must be immediate. Now, maybe we can argue whether extinction is forever or not. People are working on that, de-extinction, but this is very important that it's going to be much harder to bring species back if we lose them in their wild state. So I want to thank everyone for attending today, all of the amazing people in the leadership of the Earth BioGenome Project, too many to name. And just in the last year, all of the different funding agencies who have funded new projects. Just in the last year, you can see around the world, this is why the project is really picking up steam, because many countries are now directly funding biodiversity genomics efforts, and those efforts are all being coordinated within the Earth BioGenome Project framework. And finally, the many companies who are also working with us and contributing resources and knowledge and expertise to make all of this happen. So thanks a lot. And I guess if there's time, I can take some questions.
Thank you very much, Harris, for a fantastic presentation showing how you have progressed from the last time you presented this project here at Lewin Committee. And for sure, this is, I think, one of the most important projects in the area of genome sequence in the world. And as you said, there are many different challenges in order to accomplish this 1,800,000 genome to be sequenced by 2028. And I would like to ask you a question related to what is going on with the negotiations for the participation of Brazil and FAPESP in this fantastic project, because I think that if I am not wrong, about 25% of the Earth biodiversity sits in Brazil, and Brazil has 5 biomes that major biomes that let's say house thousands and thousands of different species. My question is, what are the difficulties that you have already faced? Because I know that you are working together with people in Brazil in order to approach the funding agencies. And what do you think that must be done in 2022. So I think that the project is in a phase. It's entering the fourth year. If I'm correct.
We finished the pilot phase, so we're really entering the first production. But yes, we're in
So what can we do to put people together in Brazil to explore the biodiversity, the Brazilian biodiversity, to contribute with this fantastic project?
Thanks, Paulo, for your question. So we've been involved with Brazil from the beginning, and FAPESP has played a very important role from the beginning. Marianne Van Sly, who is at the FAPESP headquarters and also at USP, a plant biologist there, has been involved. She's on the Earth BioGenome Project working group. She represents FAPESP on the leadership of the Earth BioGenome Project. And we even held a conference here in, I think it was 2017, which brought together researchers from all across Brazil to try and organize Brazil in a way from the bottom up, from the collection and the biobanking all the way through sequencing and analysis. And I think those efforts went very well. And what resulted was a project which I believe is in its final stages of preparation at FAPESP. There was a new call for proposals that was developed. I think that would have been released maybe a year or so ago if it weren't for the pandemic, which I think had a great effect on science throughout the world. So I'm hoping that this request for proposals will go out, because I think the basic organization that has been developed in Brazil over the last few years for this project is really still there. People want to participate, but need now the funding to bring all these groups back together in a coordinated way. That to participate in the project. And you're absolutely right, Paulo. I think the numbers are about 8%, maybe 8 to 10% actually of the world's biodiversity exists here in Brazil. So there's a lot to do here and tremendous scientific opportunities for everyone here. The thing to do is to build capacity in Brazil, build the sequencing capacity, build the human resources and the knowledge base necessary to be able to participate. And I think all the pieces really there. It's just now to be catalyzed by a funding source.
Thank you for the answer. I think that Paula has some question that was from the audience. Please.
Harris, just wondering, because I think that there are projects that are under the Ebp umbrella that are happening here in South America, so Colombia, Chile, and how in these countries this scientific community organize and get funded to launch these genome projects?
Yeah. Every country has a different, thanks for that question, great question. Every country sort of has a different history. Like in Chile, Miguel Allende there already had a very strong biodiversity sequencing program, especially looking at, Atacama, looking at the extremophiles, in those countries. So they already had a mechanism and a project. And so they were able to fold the project directly in. And then because they folded the project in, they could use that to go and try and raise more funding. Colombia is a completely different story. This is a really great success story that everyone should look at. It was very closely tied to the agricultural industry and the bioeconomy initiatives that were in Colombia. And they created an entire program from scratch. and it's called Columbia Bio. I mean, it's within the Ministry of Science, I believe. It's called one of the, or Ministry of the Environment. I can't recall. I'm sorry if there's anybody from Columbia listening, which ministry it was, but it's organized through the University of the Andes, and their vice president for research made this a central project. And then they got the government ministers involved. And they created an entire program for this, and it's called. There are two programs that are part of it: Columbia BioGenome and Grow Columbia. Grow Columbia is the agricultural component, and they're merging these two under one big umbrella project, which participates in the Earth BioGenome Project. So those are the other two major programs at this time. Mexico has a program, but that's not South America. Mexico has a program through the University UNAM, and you know, I think every country will have its own flavor on how they're to do this. And you have FPSP here, which is state; it's not really federal, so you're going to have a different kind of program that will be able to support what's happening here. The Brazilian Academy of Sciences has expressed general support for this. Also, there are people from the Natural History Museum in Rio de Janeiro that are involved and are supporting various initiatives. And also at Fiocruz, where Manuela is involved. So I think this just needs to have a source of coordination, so that, you know, everybody can work together towards some common goals for the country. And that doesn't take a lot of support, but it takes some coordination. It does. And some funds need to be devoted for that purpose.
Okay.
Right.
Very nice. I think Paula has some other questions from the audience.
Yes, in fact, it's my question. Harris, congratulations for the project. It's a very huge and ambitious project. I was wondering, what are the initiatives related to science communication on the Earth BioGenome Project?
Yeah, that's very good. So we have a communications director. It's Nicolette Caparello. There's also, I didn't mention it, but there's a communications committee. And if you look, we have a Twitter. Earth BioGenome Project has a Twitter, which is over 2,000, 2,300, 2,400 subscribers now. We broadcast every day almost a news story from one of our members. We present students and people who work. We call we are the EBP people. We broadcast about publications that come from the individual projects. We talk about some of the lead scientists. So it has been very successful and very widely read. And there's an international committee that oversees all our social media. So we have a social media presence. And then all our messaging is also coordinated by this committee at the present time. So that's how we handle our communications. We try to standardize communications, like when we are publishing this special feature in Proceedings of the National Academy of Sciences. We will coordinate with all of the institutions on the press release, for example, for this publication. So we now have a coordination mechanism that links to all of the individual institutions that are participating in the project through the communications committee. So it's running pretty well at this time, I think. And as activities continue to scale up, it's going to become a little bit more complicated, but I think we have a pretty good mechanism in place right now.
Okay, so thank you very much. I think that I have one last question. Isabel, first, please ask your question.
Okay, Harris, among the technical challenges, which one do you consider that today is the biggest one? I mean, because I have the impression, I don't know, but I mean, we have sequences, we have, you know, the DNA extraction is not great problem, but I think the assemblies,
The what?
the
Assemblies, yeah.
better, or I don't know, because
Yeah.
when I think about genomes that have high heterozygosity and repetitive elements, so I don't know how, the
Yeah.
assemblers could be better dealing with.
Yeah, so there are people working on assembly. I think 4 years ago, we weren't even talking about phased assemblies.
Yeah.
Today, it's like matter of fact. We say we're going to do a phased assembly and nobody blinks an eye. And so the assembly issues really deal with phasing and accurate, especially when you have lots of paralogs and duplicated, complicated regions, how you assemble those. So the problems that exist in assembly really boil down to those very more complicated regions of repeats and segmental duplications and ploidy issues, especially in plants that we're seeing. So those are technical challenges. Challenges also when you have single cell eukaryotes, right? How do you assemble when you sequence from a single cell if you can't grow it? This is a huge technical challenge. It's a lot of biodiversity is in single cell eukaryotes. But I still think, this issue of the sample processing, vouchering, I think it's the number one challenge. The extraction, as we mentioned, it's kind of an art. It is. It's really an art. It's collection and getting to the point of getting it to the machines is still the greatest challenge. Getting the samples, extracting high molecular weight DNA, even though two plants are the same. I mean, mammals are pretty good. I mean, there aren't too many challenges there. But when you get into the plants, metabolites, in the leaves, you use roots, you use, it seems that the greatest number of challenges exist with insects and plants, but those are being worked out. So how do you scale that so you can extract hundreds at a time, thousands at a time? This is, I think, still the number one challenge on the front end.
The high quality DNA.
High quality to keep the pipeline moving.
Okay.
And what happens when you the permitting issues? How do you get all these? If sequencing, if samples will be collected, not every country will have sequencing capacity, maybe regional. So you might have sequencing lab in Colombia and Brazil, but Guyana doesn't have it. So how do you move those samples when you have all these permitting and access in Nagoya and all of this? So we need to have blanket permits and we need to have multilateral agreements so that samples can be used for research purposes and we don't worry about the patenting issues as much. We need to get past this issue, otherwise this will go, project will stop. You'll never get to the endemic biodiversity in places of the world like in these biodiversity hotspots and where there's high endemism like Malaysia and Indonesia, and even India says, no, we're not sending samples out of the country. So we need to build that capacity in those countries so that samples can be accessed and data can be still the number one problem we face.
Okay, so my question is it's a more general question, but what the output you see for health, energy, and food security coming out of this, coming out of the EBP?
Yeah, well, that's a whole 1 hour lecture, right? Because some of that is prognostication and prediction, but we can only go on the experience that we have, from the Human Genome Project and from the agricultural genome projects. The Human Genome Project, we know, was transformational for human medicine. It's still early, but a whole new field of medicine is born, right? Personalized medicine, which came from human genome sequence, diagnostics, and now therapeutics. In agriculture, without the sequencing that has been done for the agriculturally important species of plants and animals, you wouldn't have genomic selection. This is the greatest advance in the breeding side, on the germplasm side, that's occurred. It's a revolution not having to go through the to make predictions based on phenotype rather than selection based on only exclusively on phenotype. So this is transformational for the industry. These have already happened. And so now if we need to prognosticate where things are going to happen in the future, you know, it's difficult to put your finger on one thing. But I will say in human health, it's very clear one of the applications that I talked about is prediction of the future pandemics, because through biosurveillance, and if I sequence a bat in a cave from Yunnan province, I can also capture the viruses that come with that bat. So many people who are working on this part of the project are saying we're not going to filter out and throw away the viral sequences and the bacterial sequences that come with our sample. We're going to use that information too to say these viruses are present. And then when you do, like I showed for the California Conservation Genomics Program, you do population level sequencing. Let's say in California, where hantavirus is a major threat, potential threat to human population. By sequencing 150 individuals, you can say, oh, this population may have the hantavirus. This one may not. Maybe we need to, you know, go in and control this population, not that population. So there are going to be many, I think, applications to human health through biosurveillance. I think that's going to be the most immediate. You asked about agriculture. Well, I think new sources of genetic variation for plants and animal agriculture. This is going to be, I think, come from sequencing all these wild relatives. You know, most of the agriculture in the world, I don't know what it is, some ridiculously small number of plants. I think it's something like, you know, 25 plants are used for 80% of the and of those, it's sorghum, you know, corn, wheat, and corn. It's only 4 species so we need to capture more of that variation that exists in the wild relatives to improve resistance to disease and to understand adaptation. You know, this is really going to be a big thing. You're working on adaptation. Many people are working on adaptation. And I think the alleles, the genes that we want are all out there. We just have to find them in the 99.9% of the species that we haven't sequenced yet.
So
thank you very much again, Harold, for this fantastic talk. And just to mention that in the project that Isabel and Ricardo is coordinating on sequencing the transcriptome of the Velocias from Campus Rupestris, we just ask, just for fun, we asked, are there viruses in this transcriptome? And the answer, yes. 0.5% of all the transcripts coming out from the Velociaceae encodes virus, RNA virus genome. That's really very, very, very interesting field. So thank you very much, and thank you very much for those that are attending this webinar. And I hope that Brazil will jump into the EBP as soon as possible, because we think that we have a lot to contribute for health, energy, and food security. Get in this fantastic project. Thank you very much, Harris.
Thank you very much, Paulo. And thanks everyone in the room and all of you around Sao Paulo for attending today. I appreciate your attendance. Thanks.
Close up.